Well, thank you very much, Dr. Labudi. Needless to say, I appreciate that warm introduction. I've been at surgical oncology, endocrinology, for over 50 years, and I really started, as Dr. Labudi said, at the National Cancer Institute. I actually worked in George Irvin's laboratory when I first went there as a clinical associate. George was very good to me. I learned a lot from him, both in the clinic and the laboratory. And I went back again to NCI as a senior investigator. And as Dr. Labudi mentioned, I'm back there now. When I was asked to give this lecture, I thought about historical aspects of cancer, and I was interested mostly in thyroid cancer, particularly medullary carcinoma, and I decided to really look at the significant milestones that have occurred in the diagnosis and treatment of thyroid cancer generally. Now, as I go through this, some of it might seem somewhat esoteric because I'm talking about significant things that happen. Some we use now in everyday practice, but the derivation of these and how they came about is important. And it's really about people. These milestones didn't fall out of the sky one day, somebody did it. And the people who did it had very interesting things about them, often in a relationship, sometimes complex. But collectively, these contributions kind of brought us where we are today. And I'm going to start with surgery and end with surgery. And obviously, the story begins with these two gentlemen, the upper left, Theodore Billroth and Theodore Coker. They began surgery at a very inauspicious time because the French Academy of Medicine had banned this procedure in 1950. And Samuel Gross, who was the 20th president of the American Medical Association, one of the founders and the first president of the American Surgical Association, chairman of surgery at Jefferson Medical College, made the statement that no honest and responsible surgeon would engage in it. And yet, Theodore Billroth began operating on patients with thyroid disease when he was professor of surgery in Zurich. He had started his career in Berlin at the Charité Hospital, but his most significant work on gastrointestinal surgery was done while he was in Zurich. First 20 patients he operated on with thyroid disease, 8 of them died, and he stopped operating on the thyroid gland until he assumed professorship of surgery in Vienna. His mortality rate dropped to 10%, which seems horrendous current standards, but it did. It was quite something. He was not one to shy away from surgical complications and thought in terms kind of a running morbidity mortality aspect of what had been done. And he recognized that one of the significant complications he had was tetany. As a matter of fact, one of his colleagues, Nathan Wise, reported on 3 cases of tetany at Billroth's clinic, one of whom died in the perioperative period. He was baffled by this. It was thought to be some anomaly of the thyroid gland, but remained unresolved. Theodore Coker spent almost his entire life in Bern, Switzerland. He was a very careful and meticulous surgeon, did over 5,000 thyroidectomies with a mortality rate approaching 1%. He did not have a high incidence of parathyroid problems, but he did have a very high incidence of a disease which was named cachexia struma priva, mixed edema. He was the first surgeon to win the Nobel Prize in 1909, simply described for the study of the physiology, pathology, and surgery of the thyroid gland. William Stewart Halstead, shown on the lower left, born in 1952 and was a famous surgeon in this country for many things, but he was highly interested in the thyroid gland and the parathyroid glands. Parathyroid glands, as we know them now, was not discovered until 1880 by Ivar Sandstrom in Sweden. His his observation went really unknown for several years because it was published in an obscure medical journal. When he visited both Billroth's clinic and Coker's clinic, he watched them operate and was baffled by why the occurrence of parathyroid problems was so high in one clinic but not the other. And he wrote, "I've pondered this question for many years and conclude explanation probably lies in the operative methods of the two illustrious surgeons. Coker, neat and precise, operating in a relatively bloodless manner, scrupulously moved the entire thyroid gland with little damage outside its capsule. Bill Roth, operating more rapidly, and as I recall, with less regard for tissues and less concern from hemorrhage, might easily remove the parathyroids or damage their blood supply. Charles Mayo in this country was a major force in thyroid surgery. Part of his success operating on patients with hypothyroidism was due to the multifaceted genius Henry Plummer, who administered iodide to these patients pre- and post-operatively. The same was true of Dr. Kreil, George Kreil. The last figure shown is Frank Leahy of the Leahy Clinic, also a giant in thyroid surgery, was extremely careful, paid especially attention to preserving the recurrent laryngeal nerve and the parathyroid glands. He was the first surgeon to transplant parathyroid glands to the sternocleidomastoid muscle, but the outcome of these was not known. The discovery of thyroxine was also a major advance in surgery, certainly used in patients who've had the thyroid gland removed. It was discovered by Edwin Calvin Kendall, who was head of the biochemistry unit at the Mayo Clinic. He graduated from Columbia, received his MD and PhD there, and worked for a while at Columbia. He then moved to the Mayo Clinic, and as the story goes, he made this discovery on Christmas Day in 1914. He had fallen asleep in the laboratory while an extract of hog thyroid gland had boiled and was undergoing ethanol evaporation. And this residue formed in the beaker, which contained a very high concentration of iodine. He published this in the Journal of the American Medical Association in 1915, but the definitive article was published in the Journal of Biologic Chemistry in 1927. This same year, Charles Robert Harrington and George Varga, shown below, described the structure of thyroxine, and they synthesized the compound, something that Kendall and his associates have been trying to do for several years. This is not the only thing that Kendall did, even though he's best known for thyroidologists for this discovery. He also discovered cortisone. He and Henry Showalter Hinch, shown in the figure to the left together, and in the middle, and Thaddeus Reichstein discovered this compound. They received the Nobel Prize for this observation in 1950. Thaddeus Reichstein was a very interesting person. He was one of the world's experts on ferns. And during the last 20 years of his life, he published 80 papers describing the various chromosomal complexities of the structure. Again, an example of somewhat of the varied personalities and attributes of many of these accomplished people. The discovery of TSH was critically important, and this compound is important, if for nothing else. In the last line, it promotes thyroid growth. One could almost say at the beginning, no TSH, no thyroid cancer. Of course, that's not quite true, but this compound is very influential in that regard. It's also critical for the differentiation of thyroid function, the uptake of iodine, and the excretion or secretion of I-131, of T3 and T4 into the circulation. The first lead on this was a paper by Edward Udenhuth in 1927, describing the anterior lobe with the hypothesis as a control mechanism of the function of the thyroid gland. He was from Switzerland originally, moved to the United States, and did almost all of his seminal work at the University of Maryland, where he was in the Department of Anatomy. John G. Pierce at UCLA described the alpha and beta subunits of TSH, and A.J. Van Heerle, also at UCLA, developed a radioimmunoassay for TSH. The TSH receptor was cloned at the Free University in Belgium by Gilbert Rissart and his colleagues. Each of these a major step in advancing the field. Calcitonin was discovered by Harold Kopp, University of British Columbia. It's a fascinating story because of the structure he had, this Rube Goldberg-looking apparatus to intermittently perfuse calcium and EDTA to influence the output of the structure. His target was a combination, a composite of thyroid and parathyroid gland. And when he published this paper in Nature, he described an agent that dropped blood calcium from the parathyroid glands. He named it calcitonin. At that time, nobody had any idea that the thyroid gland had anything to do with calcium metabolism. And this was accepted until Hirsch and Munson from the Harvard Dental School published 2 years later that calcitonin indeed came from the thyroid gland, and they termed it thyrocalcitonin. The name calcitonin stuck, and people use it today, and it's been used clinically mainly to track medullary thyroid carcinoma, both diagnostically and therapeutically in several fields. The person who did the most with this at the outset was Armin Tashin, and he and his colleagues at Tufts Medical Center developed a radioassay for calcitonin and also stressed it's important in the early diagnosis of medullary carcinoma, especially following calcium infusion. Bob Yeagle, MD Anderson Cancer Center, was a major part of this effort. It's puzzling what calcitonin does. And Phil Hirsch, who subsequently moved to the University of North Carolina, published a paper in Endocrine where he stated there's no readily apparent pathology due to CTX-SS of deficiency, and there's no evidence that circulating calcitonin is of substantial benefit to any mammal. So it's still baffling how this fits in the physiology of calcium metabolism, but as of today, its function is totally unknown. The development and clinical application of radioactive iodine was another major step. It began with the person in the upper left, Enrico Fermi, shown here at the Physical Institute at the Royal University of Rome. Published a paper in Nature in 1934 describing isotopes of various elements developed through neutron bombardment. This was a major advance. This work lay fallow, and he did little else for the next few years because Italy had invaded Ethiopia, and most of the money from the government went to military sources rather than research. Ernest Orlando Lawrence is best known for the development of the cyclotron, and he received the Nobel Prize for this in 1939. Glenn Seaborg, also a major force, described radioactive iodine, 128, 130, and 131. He and Lawrence were both from the University of California in Berkeley, and they both worked on the Manhattan Project. Glenn Seaborg is best known for developing what are called the transuranium elements. These are elements beyond uranium, which is 92, and all the elements prior to that are found in nature and have very long half-lives. The transuranium elements are beyond 93. They're all radioactive, have relatively short half-lives. And his group discovered 9 of these. When they started out, they had various names like neptunium and plutonium, following Uranus and Jupiter and Plato and the planetary cycle. They named some geographic areas: americium, californium, berkelium, and then several after famous people: curium, fermium, einsteinium, millennium, and nobelium. Element 103 was named for Lawrence. It's called lawrentium. Element 106 was named for Seaborg. It's Seaborgium. There's controversy about this because Seaborg was alive at the time. He was the president of the Atomic Energy Commission, but this arrested. The person in the lowermost slide is Saul Hertz. He was head of the thyroid clinic at the Massachusetts General Hospital. And one day he attended a lecture with Howard Means, who was then chairman of the Department of Medicine, and Jacob Lerman, which was given in Vanderbilt Hall at Harvard. And the topic of the lecture, given by Carl Compton, who was the president of MIT, was on the importance of physics to medicine. During that lecture, he described Fermi's work on radioisotopes. And after the lecture, they asked if it was possible to develop isotopes for iodine. Seaborg's work had not yet been done. He started with a colleague named Robley Evans at MIT to develop radioisotopes of iodine, primarily through a grant of $30,000 from the Markle Foundation. And they did develop these compounds, and Saul Hertz started treating patients, first with I-130, which has a half-life of 12 hours, and then I-131 has a half-life of 8 days. He reported on these series in the Journal of the American Medical Association. It is quite remarkable that Fermi's work was not known at the time. He went on to many other things involved in the Manhattan Project. And Hertz also treated some patients with metastatic thyroid cancer. The Atomic Energy Commission approved I-131 for medicinal use in 1946, and the first shipment of I-131 to treat a patient with thyroid cancer was actually to Barnes Hospital in St. Louis. Seaborg is the old generic of this group. He lived to be 89, but all of the others died at a relatively young age. Enrico Fermi died age of 53 of gastric carcinoma. Lawrence died at 57 following an operation at a hospital in Palo Alto. Little else is known about that. Saul Hertz died at the age of 45 of a myocardial infarct. But these gentlemen contributed greatly to the field and really brought I-131 to clinical use. For decades, people have been searching for how iodide got into the thyroid gland, and several distinguished investigators had worked on this. In 1996, Nancy Carrasco published this paper in Nature, Cloning and Characterization of the Iodide Transporter. She received her MD degree at the National Autonomous University in Mexico, moved to the United States. She worked a while at the Roche Research Institute, and then was at Einstein College of Medicine when she made this observation. It was stunning because several people had been trying to do it. She used a very high-risk technique, but she nailed this and removed all doubt about the character of the reception and its function. She currently is at Yale, where she's professor of cell and molecular physiology. Iodide is brought into the cell through this sodium iodide transporter with sodium. It's brought in as iodide, moves across the cell and into the colloid, probably helped in some way in an active process by this glycoprotein pendrin. This is named after von Pendrin of Pendrin syndrome, which he described in Lancet over 100 years ago. This is a familial syndrome characterized by familial example of deafness and goiter, and the mutations that have been found in Pendrin that account for this, described in this last paper in Nature Genetics by Eric Green's group at the National Human Research Genome Institute. While iodide is being moved across the cell, thyroglobulin is being synthesized in the endoplasmic reticulum, and it's brought by exocytosis into the colloid. It's about almost all of colloid is thyroglobulin. Iodide is oxidized by hydrogen peroxidase to iodine, and then by thyroid peroxidase, there's an organification that takes place with placement of iodine on the various tyrosol residues that reside on thyroglobulin. Thyroglobulin really serves as a scaffolding for the development structure of T3 and T4. After this conjugation, where T4 and T3 are created, there's endocytosis, then a process of proteolysis where there's cleavage of T4 and T3, and they're secreted by a somewhat unclear process into the bloodstream. The concentration of T3 and T4, or iodide, in the thyroid gland is about 50 times what it is in the bloodstream, and 80% of the iodide in our body is in the thyroid gland. Now, the sodium iodide transporter is somewhat non-discriminating as far as whether there's a radioactive iodide or whether it's normal. And it's been used to marked advantage, obviously, not only to assess thyroid function, but to treat hyperthyroidism, to ablate the thyroid remnant after thyroidectomy for thyroid cancer. And I would stay here that it should be used on a selective basis. All patients formerly used to be treated with radioactive iodine postoperatively. That's no longer the case. And it's used to treat metastatic differentiated thyroid cancer. This can be given in really equivalent doses using either thyroid hormone deprivation or recombinant human TSH. I should have probably highlighted recombinant human TSH because that in itself is a major advancement in the management of patients with thyroid cancer. There's a dark side to radioactive iodine, and I should say at the outset that thyroid cells have a limited number of mitoses. They all occur before 20 years of age. Therefore, exposure to radiation during the first years of life before the occurrence of planned cell divisions increases the risk of malignant change. And direct or incidental thyroid radiation increases the risk of well-differentiated papillary and follicular cancer 15 to 53-fold. It's at high risk in radiation at a young age and increases with follow-up. These primarily are the statements of late Elaine Ron, who was a leader in the Division of Epidemiology and Cancer Genetics at the NCI before she succumbed to pancreatic cancer at a young age. Stephen Hancock at Stanford, in nuclear medicine, was really the first person to describe clearly the occurrence of head and neck cancer following radiation to the neck. The United States, notoriously, I might add, performed a series of experiments in 1946 to 58, testing atomic weapons in two islands, Bikini and Inawak, the northernmost islands of the Marshall Islands. And in 20 of these, populations living in the lower islands were exposed to radiation. They started to develop thyroid nodules, and the United States Senate ordered an evaluation of this. And at first evaluation, it was felt that 500 excess cancers were caused by radiation exposure in the lower islands. This was later reduced to 170 of over 10,000 cancers at various sites, but they compared the incidence in this island, Rongelap, to the islands in the lower atolls. And if you look at the incidence of thyroid cancer alone, it was 95% in Rongelap compared to 12% in the lower islands. And if you march down the various organs, this is the same. This is a clear index that this was not an innocuous happening when people were exposed to ionizing radiation. This took its most graphic forms 30 years ago to the month, actually, when there was an explosion in Chernobyl. The entire roof of Unit 4 blew off and released massive amounts of nuclear fuel particles into the atmosphere, primarily I-131 and cesium-137. It was thought initially people would develop bone tumors and leukemias. In fact, this was not a major risk factor, but the amount of radioactive iodine released, immediately thought to be 100 million curies, which is an unfathomable amount. Actually, more accurately, has been estimated to be 2 or 3 billion. So this was a huge amount of release, and it was detectable in every country in the northern hemisphere. The greatest exposure to be expected was in Ukraine, Belarus, and Russia. And due to the rain and shifting winds at the time, the exposure in these sites was variable. Within a few years, there was an epidemic of childhood thyroid cancer. Most of these were differentiated, and there was a marked increase in the rate of RET fusion products, RET PTC1 and RET PTC3 primarily. The first person to discover a RET fusion was Alfredo Fusco from Naples University in Italy. Yuri Nikiforov, University of Pittsburgh, formerly a Russian pathologist, went to Russia and studied several of these children. He was one of the ones who was a major factor in understanding the expression of this disease. On the right are shown several RET fusions that occurred in these children. As I mentioned, the most common are RET PTC1 and RET PTC3. These fusions always take place at exactly the same site. They occur in intron 11. They might be a few base pairs apart, but they don't always occur at the same site, but always in the same intron. And as it turns out, RET fusions seem to be everywhere. They've been noticed in monocytic myelogenic leukemia, reported by Ballerini. Several people have reported these fusions in lung carcinoma. KF5 RET, reported by 2 or 3 investigators, CGD-C6 RET, NCOA4 RET. These don't occur very often in 1 or 2 or 3% of patients with lung cancer, almost always adenocarcinoma. But it's an example of the fact that this can occur. We've been studying a large number of patients with medullary thyroid carcinoma, trying to develop a clear picture of the driving mutations. We've looked at over 100 samples. And about half of these patients have RET mutations, and another 30 or 40% have RAS mutations, but there are 10 or 15% of patients with mutations unknown. We started to sequence these. I knew we were going to find RET fusions. We didn't. But a RET fusion was reported by Elizabeth Grubbs and her colleagues at MD Anderson Hospital. This is the first RET fusion that's been reported in medullary thyroid carcinoma, and it's a highly significant event. Ultrasound in the diagnosis of thyroid cancer is another obvious and simple milestone. It started with these 2 gentlemen, Paul Langevin and this person, Konstantin Chalowsky. He was actually a Russian immigrant collaborating with Paul Langevin. Langevin worked in the Curie's laboratory in Paris, and they were the first 2 to describe the ability by sound waves bouncing off solid objects to detect tissues or tumors or solid objects of various size in a less solid background. Their inventions and discoveries really were the basis of the sonar system used to detect submarines and icebergs, and led to great importance. Langevin actually patented this procedure. And there are various things that we use, looking at ultrasound, that try to tell whether a lesion is malignant or not. Calcification, Samoma body is one of the most reliable. Of course, local invasion of lymph node metastases, margin contoured shape, et cetera, et cetera. But this is a major advance, the first step in the evaluation of patients who have thyroid nodules. Fine needle aspiration cytology really began with Hayes Martin, head and neck surgeon at Memorial Sloan Kettering Cancer Center, when he used this technique to identify malignant tumors in patients with head and neck cancer. The technique dropped off for some reason after this initial report, and nothing much was done until 2 Swedish pathologists, Torsten Lorhagen and Per-Ole Granberg. Per-Ole Granberg's known to many members of this organization, revived the procedure and really brought it to full clinical utility. Torsten Lorhagen shown in academic regalia at a graduation at the Karolinska Institute. In the middle is Georgios Papanikolaou, who actually was right across the street from Hayes Martin at Cornell University Medical Center. There's no evidence that the 2 collaborated together, which is kind of striking, or that Papanikolaou's work never extended beyond the uterine cervix. But that is a fact. Papanikolaou moved to the University of Miami in 1961 because there was going to be a Papanikolaou Cancer Institute open, but he died a year before it actually opened. This has advanced to the point where there have been molecular technologies applied to pure cytology. These have taken the form of either shown in E.K. Alexander's work, of the Affirma technology, an RNA-based gene scanning technique to evaluate, to identify benign nodules in patients with inconclusive tumors. Yuri Nikiforov, also Sally Carty, one of the past presidents of this organization, developed a different technique based on 14 known oncogenes and various gene fusions to identify malignant lesions. I think it's fair to say that neither one of these techniques would reach the point of wide clinical applicability. They're both very expensive, and it's a question of how they're really going to fit into the diagnosis of patients with thyroid nodules. A major achievement, of course, was the discovery of DNA. This happened in 1953, this paper in Nature. It's 1 page long. It looks like 2 pages, but the second page has 6 references and some acknowledgments. And there were actually 3 papers published sequentially in Nature. This paper, there's a paper by Maurice Wilkins, who was a crystallographer at King's College in London, and the third paper by Rosalind Franklin, who also was at King's College. They opened this paper with a gross understatement. We wish to suggest a structure for the salt of DNA. This structure has novel features which are of considerable biologic interest, and they went on to describe the structure. It's an interesting story that Watson visited Maurice Wilkins at King's College. They had been discussing advances and where they were with one another. And honestly, Maurice Wilkins said he didn't have very much to tell him. He and Rosalind Franklin worked at somewhat of a distance. As a matter of fact, they had a very abrasive relationship, even reaching the point sometimes to shouts in the laboratory. Rosalind Franklin was brilliant, and she was the only one of these 4 people who had a PhD in chemistry, which allowed her probably to have a greater insight into the problem than any of the others. She was very secretive, and she did beautiful, exquisite work. Nobody took crystallography images like she did. In her paper, in Nature, she showed this figure. And when Watson had visited Maurice Wilkins, he did say that they'd been duplicating some of Rosalind Franklin's work, and she had an interesting finding. He went to the next room and got this photograph shown, this imaging procedure, and brought it back and showed it to Jim Watson. This was unknown to Rosalind Franklin. And if you read the double helix, there's a passage in there where Watson says, the instant I saw it, my mouth fell open and my heart raced. It was obvious that this was the B form of DNA, and there's no other structure that could form this but a helix. He said with a few minutes calculation, one could determine the number of rings in the structure. Whether this was the final piece of the puzzle or at what place it fitted into the discovery is unknown. Many people felt that Rosalind Franklin should have been on this paper with Watson and Crick. There's actually a Rosalind Franklin Society formed, I think partly to recognize that and also her contributions. I frankly think there's a case for that. But she did not receive the Nobel Prize with them, which was given in 1962. She died 4 years earlier of ovarian carcinoma, having worked until 2 years before her death. This is DNA. Who would have ever thought that a 5-carbon sugar, 4 nitrogenous bases, and phosphate groups would determine everything we do? How we're put together during embryogenesis, what we look like, what we think, mostly how we live, and in many cases how we die. This was an absolute major accomplishment. And when we're all gone and there's nothing left, people will remember this observation. It's really a discovery of the millennium. I don't know whose water this is, but I had to have some. Now, don't do this now, but if you go online and you look up, this is, excuse me. This is photograph 51. If you look for this online, please don't do it now. What comes up is not this photograph. What comes up is Anna Ziegler's play, which ran the entire fall season in London last year, the Noel Cowell Theater, starring none other than Nicole Kidman as Rosalind Franklin. It played to rave reviews. You might ask, what does it have to do with thyroid cancer? This is most obviously expressed in the studies of the Cancer Genome Atlas Research Network project. This is Thomas Giordano from the University of Michigan. This is Jim Fagan from Memorial Sloan Kettering Cancer Institute. He's chief of the endocrine division. And they studied 496 papillary thyroid carcinomas by SNP arrays, exome sequencing, RNA sequencing, microRNA sequencing, DNA methylation. This is a full court molecular analysis press of thyroid tumors. And what did they learn from it? There's a very low frequency of alterations relative to other carcinomas. It extended the set of known PCC driver alterations to include these 3 genes and diverse gene functions. Discoveries reduced the fraction of PTC cases with unknown oncogenic drivers from 25% to 3.5%. BRAF 600E PTCs represent a diverse group of tumors consisting of at least 4 molecular subtypes with various degrees of thyroid differentiation. BRAF-like PTCs signal preference to the MAP kinase pathway, while RAS-like PTCs deliver through MAP3 and PI3K. I could go on and on, but it really demonstrates that what's important is the molecular characterization of tumors. It's not what they look like pathologically, because there's much more to it than just the pathologic view. So most of the mutations that occur are BRAF V600E, 60%, followed by RAS, and then followed by these fusions with these illegitimate expression that would not occur without the hybrid. There are several interesting things that have come out of this regarding how these tumors behave clinically, how they respond to treatment, whether they respond to radioactive iodine or not, and this has markedly influenced the clinical management of patients with this disease. Once I saw this paper in Cell, I went down to Building 31, where the Human Genome Sequencing Project is, and told them about medullary carcinoma, how interesting it would be to do an extensive study of this tumor. And they kind of listened politely, and then politely told me that this was not a very common tumor, only 3% of thyroid cancers, and in essence told me to get lost, which I did. But I couldn't help thinking to myself, medullary carcinoma used to account for 10% of thyroid cancers until people started doing these imaging studies and finding these squeezy little innocuous micropapillary carcinomas that really messed up the deal as far as proportional occurrence of these various tumors. Now, we all know that there's been an increase in the incidence of thyroid cancer over the last few decades. You could take almost any 2 or 3 decades and you'll see this. And the critical paper describing this first came from Louise Davies at Dartmouth Medical School. She's primarily based at the Veterans Administration Hospital in Whitewash Junction, but she and her colleague made this observation that it really was due to the fact that there were increased tumors found on imaging studies. This was questioned by Amy Chen, shown here, and her colleague at Emory University, who's really the American Cancer Society of Elizabeth Ward. They felt that the actual increase was due to several stages of tumors, not just early-stage papillary carcinoma. The definitive study was done by Juan Brito and his associates at the Mayo Clinic in Olmsted County, where they studied a large number of patients. I think removed all doubt that the increased incidence is due to the fact that these tumors are found more frequently through imaging. Well, what happens? What do you do about them? Ian Hay, more than anyone else at Mayo Clinic, I think has made the case for recognizing that many of these tumors are innocuous, ought to be left alone. And a very nice study in Japan, Furuchito Ito and Akari Miyauchi at the Kuma Hospital studied 360 patients and followed them, see what happened. And the incidence of these tumors increasing by 3 millimeters is shown in this slide to the left. The incidence of nodal metastases occurring is shown in the slide to the right. So about 80–85% of these people do fine, but you can't leave them alone. I mean, they have to be followed. There's actually a paper on this meeting from UCSF that makes the case it might be more reasonable to operate on these patients, considering the cost-benefit analysis. That's the other side. There's always the other side. It's not clear quite yet how these should be managed, but I think the prevailing sentiment is that most of these should be left alone and not treated. This is the current state of the surgical management of thyroid cancer. I'm not going to spend time on this slide because it really parrots the guidelines that have been established recently by the American Thyroid Association committee that dealt with this. And I think the medullary carcinoma is very much the same, with the exception of very young children who can be operated on by prophylactic thyroidectomy. There was a report of 50 of these children operated on sequentially and then studied at least 5 years, really from 5 to 10 years after they were operated on. And we looked at calcitonin. And this was a calcium pentagastric stimulation test. This shows, say, the basal level in these 4 people and the stimulated level. In these 4 people, there's an increase following administration of calcium pentagastrin. Therefore, these patients fall out of it, they're detectable, they have residual disease. These patients who have these increases, say, example, in this patient with moderate increase, subsequently developed a very high level, added this patient, added this patient. I only make this point because you can really tell in medullary thyroid carcinoma where all the C cells are gone or not. You just measure the calcitonin and you stimulate release. There's a term used in analyzing these patients, a biochemical cure. That means less than 10 picograms a mil. I chaired a committee at ATA that established guidelines for medullary thyroid carcinoma, and I thought we were going to have a fist fight break out in the room. People who felt, biochemical cure was right, other people who felt there should be more stringent criteria. All I can say is you have elevated calcitonin, you've got C cells. It might mean nothing. These people might live a very long time with nothing else done. But the fact is that they're not cured. The last step in the advance was the development of molecular targeted therapeutics. The person more than anyone else who drove this to its endpoint was Tony Hunter. He received a PhD from Cambridge. He's now at the Salk Institute in California. And he published his paper in 1981, Epidermal Growth Factor Induces Rapid Tyrosine Phosphorylation. This seems like nothing, but it was the key to how signals get from the external environment into the cell. And they have receptors. This, for example, is the RET receptor. There's this large external ligand segment composed of 3 components that combine together. This is the RET protein to signal and turn on this internal catalytic core that has these, and all these tyrosine bases are activated and serve as docking sites for various other signaling or genetic pathways. It's very complex, but this has been worked out right along the line of the model that Tony Hunt established. The discovery of RET proto-oncogene was by Helen Dawes Keller and Lois Mulligan and their groups. And Maria Mora from Lisbon, Portugal, described RAS mutations in medullary thyroid carcinoma. It's amazing no one noticed this before she wrote this paper. But several people looked at it and said they're not there. And she found it as reported in the JCM in 2011. Fusco was the first person to describe an oncogene, as I've already mentioned. Yuri Nikiforov discovered the BRAF mutation, and Horatio Suarez from Villejuif in France and his group were the first to describe RAS mutations in patients with papillary thyroid carcinoma. The first example of a molecular targeted therapeutic in any cancer was Brian Drucker's work in chronic myelogenous leukemia with the drug Gleevec. And I'll just mention this last sentence. The estimated overall survival of patients who received imatinib as initial therapy was 89% at 60 months. 87% of these patients had cytogenetic responses, normal bone marrow. This is like something biblical. And I remember seeing the paper, and everybody thought, this is going to be easy to treat patients with solid tumors. And they instantly started to develop these compounds to treat patients with solid tumors, some of whom had thyroid cancer. George Dimitri is known for his work with the first solid tumor, was treated with Gleevec, which is a GIST tumor. Shown here in a CT scan. This is the PET scan. 1 month later, this tumor disappeared. These were all what are called partial remissions. They're kind of gone, but they all come back. That was not true in Brian Drucker's work with CML. Charles Sawyers at Memorial Sloan Kettering Cancer Center and his group really developed a theory for combinatorial therapy for CML. Hagot Kanarjian from MD Anderson Hospital really was the first person to apply this dual therapy in patients with CML with a substantial remission rate. It was quite remarkable that some of these patients had such a remission. 11 or 12 patients with a chronic phase had a complete hematologic remission. Some of the ones with advanced blast crisis did not do as well. Now these are what are called waterfall plots from clinical trials. And they're great in a way because you can get an idea on every patient in the trial what the maximum response is. That is, how much of a tumor reduction you get or how much the increase is, whether they respond or not. The problem is that you don't know how long the response lasts. I mean, you take this picture right here, say, 2 months later, the shades might go up, and there are not that many patients who still are in response. But it does give you a very good idea of what one can expect. I want to come back to this image in a moment. This is data from the recent Decision trial at Marcia Bros. This is Axitinib trial, Ezra Cohen, University of Chicago. This is Motacinib trial, reported by Steve Sherman, New England Journal of Medicine. This had only a 13% response rate, and Amgen, who produced this drug, took it off the market shortly after this paper was published because of the low response rate. This is Beth Fox's data, children with MBN2B treated with Vandetanib. This is the original data from the XL184 trial, or Cabozantinib. And this is the data from the Pazatinib trial, Keith Bible at the Mayo Clinic. These are the results of the only drugs that have been approved by the FDA to treat advanced thyroid cancer. These two, Sorafenib and Avandanib, to treat patients with differentiated thyroid carcinoma, and Cabozantinib and Vandetanib to treat patients with medullary carcinoma. You tend to look at these trials and you try to draw a conclusion from looking at the trial and reading the conclusion in the paper. You can't do it. These trials are designed differently. They have different eligibility criteria, different endpoints. And it's, unless you really read the papers carefully and compare one to the others, it's really difficult to come away with a conclusion. All of the remissions that occur in these patients are partial. There are no complete remissions. There are 2 or 3 reported in the Lenvatinib trial, but they were early. Now, I think we're at a stage with this right now where it best be described by a comment that Lewis Thomas made. People in this room are too young to know who Lewis Thomas is. He was a dean at Yale Medical School. He was later president at Memorial Sloan Kettering. He wrote a series of articles for the New England Journal of Medicine, quarterly articles from 1970 to 1973. They were called Notes of a Biology Watcher. And in one of these, he wrote about technology in medicine. He coined the phrase halfway technology. It was to describe things like kidney dialysis or the iron lung, where you have a technique that's not curative, but it carries the patient along during a very prolonged period where they can be sustained. That's what we're doing with these clinical trials. It really is the halfway technology because this is not curative for these patients. And what will happen only rests with the discovery of either new compounds or the development of combinatorial therapy. I'm not going to go into this slide, but it's here for a purpose. And there's been a sentiment that you don't treat patients with a certain type of tumor. You treat the mutation. Now, this shows 3 different clinical trials on people who all have the same mutation. It's the BRAF mutation, V600E. It also uses the same drug that initially showed great promise with melanoma, vemurafenib. As a matter of fact, they showed overall survival advantage with this drug compared to placebo. In patients with colon cancer, it does nothing. And it's due to the fact that these patients have a signaling that's of epidermal growth factor that causes their resistance. This paper was reported by Marcia Bros, International Endocrine Meeting, and there was a modest increase in patients, a modest advantage in treatment of patients who had treatment with this drug, but it's nowhere near what one sees with melanoma. All this says is there are lineage differences in these lines. It doesn't just depend on mutation or the drug being given, which are both the same, but the differences in the tumor. This is demonstrated in this slide here about Beth Fox's data published in 2013 of children treated with vandetanib, all of whom had MEN2B and the M918T mutation. Same mutation, same drug. Look at the difference in response in the patients. Now, what this is due to is unknown, but again, these tumors are different. There's a much more complex matter going on than we think. So what's ahead for thyroid cancer? Development of new operative technology. This is a technique to remove the thyroid gland going transaxillary approach, which hard for me to understand how you would safely do a total thyroidectomy and remove all of the lymph nodes, but it's done. Or take this technique where the thyroid gland is approached transorally. It seems like the bravest of all of these approaches. And when I was editor of the World Journal of Surgery, I received this paper from this author, Transverse Submammary Skin Incision for Thyroidectomy. I kind of looked at it and I sent it out for review. It was rejected, but I wish I had published it because it was from an author in Nigeria, and blacks develop keloids frequently, and there's a real taboo in certain cultures in Africa where a scar on a woman is really taboo. Oftentimes they're not taken for a mate. So this surgeon did submammary incision, would reflect the entire breastplate up over the face, remove the thyroid gland. He had pre and post-op pictures. They were very impressive. This is just a short article published in the British Journal of Surgery. It didn't show any photographs. But again, it's an example of expansion of technology. One wonders, however, how far this can go. There need to be more specific molecular targeted therapeutics and the development of immunotherapy for treating cancer, which I hope Steve Rosenberg will talk about. I've never done this before, but I want to spend a minute talking about people who were in our group who've worked on thyroid cancer over the years. Many of them are members of the society, and many of them have made significant contributions. Jim Howe, Terry Larimore, and David Chi all worked with Helen Donis Keller and Paul Goodfellow to clone the RET proto-oncogene. Helen Donis Keller was received a PhD from Harvard with Wallace Gilbert. She was the first person to develop a genetic map, and she, as Francis Collins, got so far in front of everybody else that it was difficult to imagine someone being able to compete with him. She was recruited at Washington University, as was Paul Goodfellow. She was the lead author of the cloning of the RET gene causing MEN2A and FMTC. Michael Skinner operated on most of the patients prophylactically, was the lead author of the 2005 paper. Reporting the results of this, John Fay was also in the group. He's currently at Ohio State University, integral part of the program Project Grant, and also the SPORE grant on thyroid cancer. One of the two funded the other at Memorial Sloan Kettering. See John Olson, Kluge Professor of Surgery at Maryland, Vice Chairman of the department, Vice Chairman of the Greenbaum Cancer Center, worked with Helen Donis Keller on MEN1, has maintained an interest in parathyroid disease, published a very nice paper in Virginia National Academy of Sciences a year ago on genetic and functional studies of isolated parathyroid cells, parathyroid tumors, explained the complex pathogenesis of parathyroid neoplasia. This is Will Galanders, who's professor of surgery at Washington University, currently head of the training grant interested in breast cancer. This is Mike Brunt, first trainee on the training grant. He described the initial reaction of short-term infusion of calcium in instrument lease, first reported by Ed Kaplan in 2-year infusion of calcium some years before that. Steve Kim, professor of surgery at Carmona's Cancer Center. It's Rebecca Aft, who developed a very novel technique to detect isolated breast cancer cells in bone marrow using a multiplex gene expression method. This is Ruth Decker. She described the relationship of Hirschsprung's disease and medullary carcinoma and its genotype-phenotype relationship to exon 10 mutations, reported in 1988. in human molecular genetics. John Yims, professor of surgery at City of Hope. Jeff Norton, known to many people in this group, had a major force in MEN1. He worked with Steve Rosenberg, was in his branch, head of the metabolic section. He and Robert Jensen wrote these seminal papers on the management of patients with MEN1. He's currently the Ellenberg Professor of Surgery at Stanford. To top it all off, he prolonged Steve Jobs' life by removing his pancreatic tumor. This is Marston Linehan, interested in urology, was in our group. He currently is chief of urologic oncology branch at the NIH. He and his group define the molecular characterization of kidney tumors. Gerald Doherty, immediate past president of this organization, head of video endocrinology, a leader in the American Thyroid Association. He's currently the James Utley Professor at Boston. He was formerly the Norman Thompson Professor, Division of General Surgery at Michigan. Bill Kantz, interested in focal adhesion kinases, formerly chair of surgery at Florida, chairman of surgery at Roswell Park Memorial Institute. Jeff Moley, a longtime colleague, clinical surgeon, professor of surgery at Washington University. And Stan Ashley, who's currently a Sawyer professor of surgery at the Brigham, chief medical officer at the Brigham Hospital. He was in the laboratory with Daniela Gelhart, working on MEN1. He developed hyperthyroidism, was treated with I-131. He was persona non grata in the laboratory because he was radioactive and he messed up everybody's calculations, so he had to go sit on a bench and do theoretical work the rest of the time he was in the laboratory. We had several postdoctoral fellows from abroad. This is Kwazo Shimazu, chairman of the Department of Surgery at Nippon University in Tokyo. This is Derek Alderson, working on islet cell transplantation with Paul Lacey. He's chairman of the Department of Surgery at the University of Birmingham, current editor of the British Journal of Surgery. The late John Farndon described FMTC, chairman of surgery at the University of Bristol. This is Martin Backdahl, also worked on MEN1, chairman of the Department of Surgery at the Karolinska Institute, Stockholm. This is Jordan Wallin, interested in anaplastic thyroid carcinoma. He's chairman of the most recently created medical school in Sweden, Örebro University. This is Detlef Baric, chairman of surgery, Marburg University. He described BRCA1 mutation in patients with familial pancreatic carcinoma. This is Klaus Herfurth, interested in parathyroid disease. His father was chairman of surgery in Heidelberg, Christian Herfurth. He didn't want to follow his father's footsteps. He's now professor of radiotherapy at the Karolinska. And this is Thomas Musolt, who's chief of endocrine surgery at Mainz. Most recently, we've worked with several investigators that have been interested in Drosophila. Renee Reed here is the person who really developed Drosophila model for medullary thyroid carcinoma. She's currently in the Department of Pharmacology at Emory. This is Rolf Kagan, who's head of developmental regenerative biology at Mount Sinai. This is Paul Meltzer, the chief of the endocrine branch where I work. This is Beth Fox, currently associate professor Penn, mainly at Children's Hospital. This is Massimo Santoro. He has the keenest insight to medullary carcinoma of anyone. He's at Naples. There's two postdoctoral fellows that worked with us. Anita Tamburino, she described activation of the mTOR pathway in MTC. And this is Paulo Serrero. I mention these people for two reasons. One is to recognize what they've done, and also to tell how important it is to spend time in a basic science laboratory. I think there's been a drift in American surgery away from this, and I regret it. There's never been a more exciting time in cancer research, in endocrine cancer research. I said that 5 years ago, I said it 10 years ago, I said it 20 years ago. Every day, your patients are trying to tell you something about their disease process. They're also asking something. What's happened to me? Why am I sick? Can you help me? If you will take your patient's problem to the laboratory or learn to collaborate with basic scientists who know the field, and there are ways to collaborate with basic scientists, you have no idea how much you can accomplish in understanding the patient's disease process and relieve their suffering, but it does take time and persistence and patience. Everybody finishes a residency and a training program owing money. They have families, a lot of responsibilities. They're eager to practice their specialty. And there's a risk in going off in a laboratory. But sometimes it's important that you live part of your life without a safety net. And there's a great set of opportunities out there. This organization could have a franchise on endocrine tumors from the standpoint of doing clinical trials. Even if you just develop annotated tissue banks, you have no idea how valuable that is to the oncologic community. But my hope is that you won't leave much on the table. It's an honor for me to give this lecture. Thank you very much.